Hi, this video will talk about the cell cycle, more precisely, the eukaryotic cell cycle. Firstly, I would like to point out that this model would be best represented using a 3D material. However, due to coronavirus and social isolation, I wasn't able to purchase this material. Therefore, I had to improvise using a 2D model. Okay, firstly, the cell cycle is a sequence of events between one cell division and the next. It's basically the stages in which the cell grows and prepares for division and eventually divides. It has two main phases, the interphase and the cell division. But what is a cell? The word cell comes from Latin, meaning small room, and is defined as the basic structural, functional, and biological unit of all organisms. It is the smallest unit of life. But what do we find inside a cell? Surrounding the cell is the cell membrane, and within the cell we find the nucleus. Around the nucleus we find the endoplasmic reticulum. Then we have the surrounding area inside the cell, which is called the cytoplasm. The cytoplasm contains various cell organelles, like the mitochondria and the Golgi apparatus. Most importantly, we find the centrosomes, which are extremely important in cell division. They help in separating the cells with its microtubules. Within the nucleus, we find the genetic material. Genetic material is basically DNA, and DNA is a double helix molecule made of two antiparallel strands of nucleotides, or nucleic acids, linked by the hydrogen bonding between complementary base pairs. Then we have the centrosomes, which are a thread-like structure of nucleic acids and proteins found in the nucleus. This one here is made up of two sister chromatids, which are genetically identical and are held together by the centromere. Therefore, we call it a duplicated chromosome. Humans have 23 pairs of chromosomes. This is a drawing which might help you understand better the chromosomes and their structures. Most of the cells in our body divide continuously, but the cycle in which it prepares for cell division and actually divides into two daughter cells is called the cell cycle. Firstly, we have the interphase, which is a very active phase in the life of a cell when many metabolic reactions occur, such as DNA replication in the nucleus. Secondly, we have the cell division phase, where the division process itself takes place. The division process in this case is called mitosis. Zooming in the interphase, we have three main stages, which are called G1, S, and G2. At the very beginning of interphase, there is one daughter cell, and in its genetic material, we find chromosomes. In this model, I will start with a diploid cell, which has 6 chromosomes. The red represents the chromosomes which come from their mother, and the green come from the father. They are homologous chromosomes, meaning they have the same length but don't do the same thing. Oh, and also I wanted to point out a major limitation of this model, as humans would have 46 chromosomes and not 6. The daughter cell will enter the first phase, G1, known as gap1, and the cellular content will start to grow and prepare for DNA replication. All proteins necessary for the division are synthesized, but the chromosomes will not yet start duplicating. At the end of G1, the cells will still have the same amount of chromosomes, 6 chromosomes. Then the cell will enter the S phase, the synthesis phase. Throughout the S phase, the DNA is replicated and chromosomes will duplicate, also known as chromosome replication. Each chromosome duplicates. The chromosomes grow from being a single chromosome with one chromatid to a duplicated chromosome with two sister chromatids, so that after cell division, both daughter cells will contain a complete set of genes. At this point, DNA and the cell content has doubled, but the number of chromosomes remains the same, 6 chromosomes. However, it has doubled the amount of chromatids, therefore 12 chromatids and 6 chromosomes. After the S phase, the cell will go on to G2 or gap 2. This is where the cell finishes growing and prepares for cell division. Several additional biochemical events for the cell division takes place, making the cell ready for cell division. It also consists of many checkpoints, which the cell examines internal and external cues and decides whether or not to move on with cell division. At the end of G2, the cell will still have 6 chromosomes and 12 sister chromatids. This is the end for interphase. Therefore, now the cell will enter the cell division phase and go through mitosis. Mitosis is a type of cell division in which one cell, the mother cell, divides to produce 2 new cells, the daughter cells, that are genetically identical to itself. Overall, mitosis is the part of the division process in which the DNA of the cell's nucleus splits into 2 equal sets of chromosomes. Fun fact, eukaryotes need to use mitosis in cell division because they have a nucleus that contains chromosomes, while prokaryotes don't contain a nucleus, therefore do not use mitosis for cell division. Mitosis consists of 4 basic phases: prophase, metaphase, anaphase, and telophase. But some break prophase into 2 phases: the early phase called prophase and the late phase called prometaphase. These phases occur in a strict sequential order, and cytokinesis, the process of dividing the cell contents to make 2 new cells, happens simultaneously with telophase. In early prophase, the cell starts to break down some structures and build others up, setting the stage for the division of the chromosomes. The centrosomes start to condense to form a compact meiotic chromosome, making them easier to pull apart later on. And the nucleus, or the nucleoli, plural, a part of the nucleus where the ribosomes are made, disappears. This is a sign that the nucleus is getting ready to break down. The centrosomes start to form the meiotic spindle. The spindle is a structure made of microtubules, strong fibers, which some say are part of the cell's skeleton. Its job is to organize the chromosomes and move them around during mitosis. The spindle grows between the centrosome as they move apart. In the late prophase, sometimes called prometaphase, the meiotic spindle begins to capture and organize the chromosomes. The nuclear envelope breaks down, releasing the chromosomes, and the chromosomes finish condensing, so they are very compact. The meiotic spindle grows more, and some of the microtubules start to capture chromosomes. The microtubules can bind to the chromosomes at the kinetochore, a patch of protein found in the centromere of each sister chromatid. In metaphase, the centromere regions connecting paired chromatids become aligned in a plane at the cell's equator. At this stage, the 2 kinetochores of each chromosome should be attached to the microtubules from opposite spindle poles. Basically, the microtubules are attached to the centromere. The microtubule spindle fibers will contract, causing the chromosomes to line up separately along the center of the cell. The equatorial or metaphase plate. Before proceeding to anaphase, the cell will check to make sure that all the chromosomes are at the metaphase plates with the centromeres correctly attached to the microtubules. This is called the spindle checkpoint and helps to ensure that the sister chromatids will split evenly between the 2 daughter cells when they separate in the next step. If a chromosome is not properly aligned or attached, the cell will halt division until the problem is fixed. In anaphase, the sister chromatids separate from each other and are pulled towards the opposite ends of the cell. The centromere pairs from each chromosome separate, and the single chromatids, which are now considered individual chromosomes, are pulled towards the opposite ends of the cell. As they are pulled to opposite ends of the cell, the spindle fibers start to contrast. In telophase, the cell is nearly done dividing and it starts to reestablish its normal structures as cytokinesis takes place simultaneously. Once the two sets of identical chromosomes reach the poles, the meiotic spindle is broken down into its building blocks. Two new nuclei form, one for each set of chromosomes. Nuclear membranes and nucleoli reappears. The chromosomes decondense and return to their form. Simultaneously to telophase, cytokinesis happens, dividing the cell into two daughter cells. It is important to note that cytokinesis is different in animals and plants. In animal cells, cytokinesis is contractile, pinching the cell into two. Overall, the plasma membrane is pulled inwards around the equator of the cell to form the cleavage furrow by using a ring of contractile proteins. When the cleavage furrow reaches the center, the cell is pinched apart into two daughter cells. Then, cytokinesis is finished, and we end up with two new cells, each with a complete set of chromosomes identical to those of their mother. In this model, meaning that each diploid daughter cell contains 6 chromosomes with one single chromatid. The cell cycle is now completed.